Okay, so good morning. My name is Simon Anders. And welcome to this lecture, which was a plan of a while ago to do this, and now ended up to be a little bit different than planned, and we'll see how it goes. Originally, the idea was to offer it mainly for students from the math and computer science department, but my guess is that most of you are from the systems biology course, right? And background was they had some serious administrative troubles in the math department, and so didn't announce the lecture because the only person who knows how to announce lectures left for vacation without telling anybody. Morning. So we now this year. The plan is to tell you a bit about single-cell omics. And I thought I'll start by pointing out, so you have to give me a moment to get into it. It was at the beginning of a term. And yeah, my brain also has to switch back to lecturing mode. So the essential idea behind this lecture was a bit the observation that in bioinformatics there's actually two meanings of the terms. And curiously, many people, especially coming out of biology, only realize this difference too late. There's on the one hand, whereas bioinformatics as how do you apply bioinformatics tool to analyze your data, but of course there's on the other end, how do you make bioinformatics tools? How are they actually programmed? And if you look at all these bioinformatics tools that most of us are using, they are typically produced not by biologists, but by physicists and mathematicians. And the reason behind this is simply that your typical biology studies seem to skip over everything mathematical. Which brings us to a big issue because, of course, so I'm a physicist myself. It took me a long time to learn enough biology to be able to successfully pretend that I know what I'm talking about when I do biology. On the other hand, so we physicists also don't really know enough about biology, while our usual biology lecture or curriculum doesn't give you enough math background to actually do anything to work with big numbers. So somehow we have a gap here which somehow needs to be bridged, and at the moment it's usually bridged by people like me or so, who after their postdoc spent a lot of time trying to figure out the other side. Biologists who spend a lot of time learning math and so on, but it seems to be an ironclad rule that we do this only after the master, because we don't get it in the curriculum. So this is now an approach to do it already now, which I think is seriously is important. And the question, of course, which I've discussed a lot with colleagues from both sides, about what works better, teaching biologists the mathematics or teaching mathematicians the biology. Who knows? But now we ended up, yeah, you're looking for, I am. And now we are, where was I? And now I think we ended up being a quite mixed audience, so which might be not so bad, because in the end we can fill each other in. So, but still, what I really want to do is the aim of this lecture is not giving you the standard cookbook recipe how to analyze your single cell data, which might come out of your experiments, but rather understand what's actually going on under the hood of all these tools, how we do this ourselves. So my typical idea how to do this is, I think now we will start off by just using, trying out one of the standard analysis pipelines, and we will rush through it once today, and we will always at each step, we will wonder a bit, okay, this has a nice fancy name, but what's actually happening there? And in the following weeks, we will dissect all these individual steps and try to understand what's going on under there and what's the math behind it. In the end, the whole math isn't that difficult. The level is essentially you need to know a little bit of linear algebra and vector calculus and so on. And we'll see how deep we have to go into this introduction or not. So maybe let's start off. I would like to make a quick round where each of you tells me which side they are from. So what are you studying? What's your exposure to biology, to programming, and to mathematics, respectively. So, as I already said, I'm a physicist. I switched after my PhD to biology and been trying now since a while. And by now, I've sort of managed to pretend that I'm an actual, that people usually need to look twice, that they notice that actually I have no clue about biology. And physics is, I always like to say, the science of applying mathematics to understand nature. And that's what we're doing here when we try to use mathematical methods to understand biology. So on one hand, we can say we're doing physics here. On the other hand, of course, we want to try to understand gene regulation and things like this. So we are primarily in biology. But let's go around. So let's start with you.
Diana, I'm a postdoc at the moment in mathematics, and I work with models of biology, so I'm somewhere in between.
So I'm a first year master's student in systems biology, and I had a bit of previous experience in mathematics.
So I'm a senior scientist in geometry and topology. I recently, a couple of years, got interested in topological data analysis and applications to biology. Already learned a lot from personal discussion with Simon, and I hope to learn even more here in this lecture. Totally new subject.
Hi, I'm Nida, and I'm studying Masters in Scientific Computing. My application area is biology, so worked in DCAP set, but it was mostly for optimization problems, and I started my thesis at the Unique Clinicums. Yeah.
I'm Michelle, I study systems biology in the second year, and my programming experience is mostly in R, just basic. And we had a math lecture, but it's also very basic, so
I'm Kathleen. I'm also a second year master's student from systems biology, and I think I have similar experience to Michelle. My bachelor's was more biological.
You did your bachelor's here, or?
I don't know.
Okay. I also study systems biology in the second year. Same. Michelle, I do some art and a bit of Python too. Right now I'm doing a practical inducing this omics analysis, so it's cool to learn a bit more.
Hi, I'm Katarina. I'm studying neuroscience in the third year now. I'm a master's thesis at the moment, and I actually switched from wet lab to dry lab, so I've been doing bioinformatics for the past 1.5 years. So, yeah, I've worked a bit with things.
I'm Philip. I'm a PhD student in the group of SaaS colleagues at the Uniclinicum, and I also had a previous internship with Simon some time ago, and I studied biochemistry originally, and then went to the dry lab 2 or 3 years ago.
I'm Prashad. I'm also a master's student in scientific computing. I am mostly interested in PDEs and vision first problems, so I'm a bit far from biology, but I am doing my thesis in mathematical oncology group.
I see. Okay, if I made it correct, we have here 2 mathematicians, 2 scientific computing, and 6 biologists. Good mix. Let's see. Okay, what I thought doing now is let's first, I will probably often have to ask who knows what already, because we are such a different thing. Who knows how single-cell omics works in principle and what this is all about? Okay. Then we need to quickly summarize for those 2 which aren't. So I apologize that this lecture isn't that well prepared, simply because I didn't know what to prepare, because you're also separate. And now the biologist can sleep for 5 minutes while I explain to the rest what omics and so is all about. So the fundamental thing what we are trying to do here is essentially to understand what's going on in cells. And the way how I usually like to think about is if you, all your cells have the same DNA, but they do a very different thing depending on whether it's a skin cell or a heart cell or a muscle cell, and whether they are happy or diseased or just reacting to something. And so in a way, mathematically speaking, cells are in different states even with the same DNA. And sometimes the states are so different that we say this is a different type of cell. And we want to understand what cell states and types are in our tissue sample. And for this, we need something which is some easy way of getting lots of information from our cells. And the easiest to do is RNA sequencing, because you all know the basic information flow in the cell, according to the so-called central dogma of molecular biology, is the genes which are active gets transcribed into RNA, the RNA gets out of the cell and either does some regulation or, most importantly, gets translated into protein. And then, of course, so if you want to describe a complete state of a cell, we would have to explain which genes are active, which proteins are translated, which proteins are currently in the cell, where do they sit, what do they do, what PTMs do they have, etc. And we have lots of different lab techniques to get all this. But the one thing which is easiest to get is RNA, because we can simply extract all the RNA and sequence it. So if our aim is to get as much information as possible, so mathematically speaking, a high-dimensional information vector out of one cell, then the easiest is to extract the RNA and sequence it. And we can reasonably hope that if two cells are in a different state, then it could conceivably be the case that they have the same, that they contain the same RNA complement, but differ only in what proteins they are, but practically that won't happen. There will always be some difference in the RNA. So the abundance of a different RNA species is a good proxy for the cell state. And what we will do a lot in the following thing is we will describe each cell by a vector where each element tells us how much RNA we have from this gene. So we will speak about something which we'll call the feature space or the gene expression space, where which is a vector space, and the vectors in it describe always one cell, and each element tells us how much RNA we have from this specific gene. So each gene in the genome is represented as one dimension in this feature space, and our feature vector is just a list of numbers which tells us for each gene how strongly this thing is expressed. And the reason why we like to do this is because we have these single-cell sequencing techniques which have become really mature in the last 4 or 5 years, where we can take a few thousand of cells from a sample, dissociate them so that they are all suspended in some liquid, put them into this 10x sequencing machine, and sequence them. And the machine that everybody uses, let's take here a 10x Chromium. Let's look at one of the typical advertisement pictures. Yes, the picture everybody always looks at. See if I, come on, open image in new tab, maybe here. We have a test tube where each cell is suspended as a single cell hovering in some suspension, in some liquid, and when we put it into a microfluidics machine where the cells come along here from below, and from the side come silica beads, which have DNA primers on them. And when you have this oil, this water in oil droplets, where you have a small water droplets that typically will contain one of these beads and one cell. In practice, of course, what we have here is a Poisson distribution. The number of cells and the number of beads which end up in each droplet depends on, it will be Poisson distributed, and most of the droplets will be empty, but in the end we only use those droplets where one cell and one bead came together and throw away the rest. And what we have there is now, what then happens in these droplets is, do they have this? Oh no, we don't have this. Then I have to simply draw it on the board, and I have to describe it verbally for those people who will then later listen to this recording. So on these beads, let's ask who already knows how 10x works. Nobody, okay. Then let's talk about it. So our aim is that afterwards, let's go the other way around, who knows how RNA sequencing in bulk works? So, okay, for those who don't, if you have somehow a suspension with lots of RNA inside, and you want to sequence that, what you do is that you have your RNA species, you ligate some adapters to both ends with a known sequence, and then you put it in these high throughput sequencing machines, these Illumina, HiSeq or whatever they are called. And on this machine, you then have, let's quickly get a picture for this as well. 10 Illumina rich PCR images. Here's a nice picture. And play here. Open image in new tab. Okay, you have your DNA fragment, so you take your RNA, you reverse complement it into DNA, and then the transcripts from each gene gets broken up in smaller pieces and transferred from RNA into DNA with reverse transcriptase. So sorry for the mathematicians to occasionally now just use biology stuff. You just have to ask if you're unfamiliar with it. And what you now have in this Illumina sequencing machine is on the ground. You have a little chip called a flow cell, which at the bottom has all these little DNA pieces attached to it, which have two fixed sequences. And in our library preparation, we attach these kinds of so-called adapters to both ends to it with some chemical ligation operation. And then if I pour it over with flow cells, then they will attach to it. And it's actually drawn only here on this picture how it really looks like is. Here are these primers, which are attached to it, and now comes a sequence, which here has, maybe we even have color stuff, yeah. Which here has the sequence which is complementary to that one. And so the two base pair ends just attached to that. And then on the other end we have this side here, and some of these things are purple and some are orange. And what then happens is later we then do a polymerase chain reaction, and the PCR, as you know, just starts like double-stranded and elongates, and then we have here now a copy of this thing. This copy will now bend over, because here at the beginning we have this purple, so it bends over and attaches to that one. Because at the end it has the purple sequence, so it base pairs to the purple sequence which is attached here, and then with our next PCR, this thing will again produce something like this here with some orange stuff. And in the end, you can imagine every time I make a new sequence, it's attached at one end because it grows out to attached primers, and the other end will then bend over and will find another of these similar of the other primers attached to it, and in the end you then have each initial sequence grows a whole cluster of a few thousand copies, which are all at the same spot because it can't travel far, because all the sequences are, of course, within the reach of the individual DNA fragment. And so in the end, if I put a few billion, if I put, say, 100 million DNA sequences on my flow cell, then they will spread out. Each one will attach somewhere else, and around each one will grow a little cluster with a few thousand copies. And now I can do sequencing by synthesis, so standard Sanger sequencing, where I always attach individual fluorescently labeled bases and look with a microscope to see what I've attached, because at each individual point another thing will be attached, and at each individual of these clusters here different bases attached, which has a different color of fluorophore. And if you look under the microscope, then I see different colors popping up here, and I see what base is in each of these things, and that I do until I have sequenced the whole thing. So I get a stack of images which tell me the sequence of each of these clusters. In the end, my machine gives me a long text file which just contains these sequences. Each row is sequence. Usually they are shown as something called a fast tube file. That means there will always be for each read So we call this a read, what we read off from one of these clusters. And for each read, we get we then get three pieces of information, namely the actual sequence, where the flow cell the thing was detected, which is usually a sequence we have but don't really use because it's not very useful, and how sure the machine was that it has sequenced it correctly. So it will indicate with a bad score if colors have been blurred, or maybe two clusters were too close and couldn't quite be distinguished, and so on. So this is this high-throughput sequencing technique, which was developed in the early 2000s and has become better and better, so that now for a few €100 we can sequence billions of DNA fragments to either sequence the whole DNA of a person, or if we have RNA, we reverse complement the DNA and sequence all this. And usually if I now have one big if I have now a sample and I sequence, I want to do RNA sequencing from, let's say, a skin biopsy or something like that, when we will see all the RNA which was in there, because in the classical workflow we lyse the cells, so the RNA of all the different cells gets together and cannot be properly distinguished. So that if I now compare, to take a typical example, let's say a bit of a skin sample from healthy skin with skin sample from skin cancer, and I want to see what's changed, then I will see that that certain reads become more abundant than others. So that I suddenly see there is more transcripts of gene X in the cancer sample than in the healthy sample. So obviously gene X has something to do with the cancer. But what we don't know is why gene X has gone up. Has gene X gone up because the skin cells turned into cancer cells and now we get more of them, or has gene X gone up because the immune system has noticed the cancer and infiltrated it, and actually this gene X is not something that's typically of cancer cells, but it's something that is typically of immune cells. People always like to compare this with, there's always this picture of, I don't have it here now, a single cell fruit smoothie. That's the one which you have to keep in mind, because when people like to talk about single cell sequencing, they always say standard bulk sequencing is like a fruit smoothie. You get all your, you have a sample of a fruit salad, but instead of picking out which fruits are in the salad, you make a smoothie and just check how the color has changed. What we actually want to know is if a gene changes, is this because we still have the same cells in our other sample, but in the cells, this gene is now expressed more strongly, or is it that type of cells which expresses this cell more strongly has gone up? So it could be that the gene is specific, that our sample is a mixture of different cell types, and the gene we are looking at is specific to one of these cell types, and the number of these cells has gone up, rather the expression strength of this gene within these cells, and we can't distinguish that. This is why we want to record of any two reads, whether they come from the same sample, whether they come from the same cell or not. And once we've done that, we have a much richer information. So how do we do that? This is where the single cell sequencing comes in. Here it is, where we have this clever bead. Here's the bead, but I draw a new one. So, see if I get this correct. We have these, as you remember, we fuse in each droplet the silicon beads with cells. The droplet also contains some detergent to lyse the cell wall so that the RNA gets released and comes in contact with these silica beads. And these silica beads, they are coated with small pieces of DNA. And each individual piece looks as follows. Here is our bead, and on these things we now have here it ends in a lot of teeth. Now the biologists, so 2/3 of you will know that most messenger RNA is polyadenylated, so it has a tail of only A's at the end. So each messenger RNA will stick to this teeth with its poly A tail, and after the poly A tail comes then the actual end of the transcript. It's the untranslated region. And then comes next here 3 different pieces, namely the sequencing adapter. Is it drawn here? Ah, yes, here it's drawn. The barcode, cell, barcode, and the UMI. Now, the way how we make these beads is very clever. They synthesize the DNA on the bead, and first they synthesize on all beads the same sequence, namely the Illumina sequence that we later need to have at the end of our beads, of our transcript, so that we can actually sequence them. Next comes the cell barcode, which is one different, which is the same over all the beads within. It's the same over all these different things in the beads, but it differs from bead to bead. And last comes the unified molecular identifier, which is the same over, which is different between each of these little pieces here. So this here is constant. This here is constant across a bead. And this here is completely variable. So if I go from one bead to the next, the cell barcode changes. But if I go from one of these little things to the next within a bead, the cell barcode stays the same, but the UMI changes. What's the point of this? The point is that we can later take, if we have a read, because if we went sequence that, you can already see where this is going. We will do a polymerase which amplifies, which elongates this, copies that, and in the end we get here this finally library construct. Let's read it from the left. There is the Illumina adapter and the read adapter, then comes this cell barcode, the UMI, and then all the T's. And if you read it if you read the other end of the transcript, we see this little piece of the end of the transcript molecule. The read to the end of the transcript molecule allows us to identify which gene it was, because we just search for this sequence in the genome, and then we know where in the genome this transcript comes from and identify it. If we come from the front side, we will see the 10x barcode, which tells us which cell it was, because if we see 2 reads which have the same barcode, we know that they are from the same bead. And because they come from the same bead, they have been the same droplet, and if everything worked correctly, so that we had only one cell per droplet, they should be the same. While the UMI should really differ from each cell to the next, because, from each transcript molecule to the next. If we see 2 reads with the same UMI, then this would mean that these are both PCR duplicates of one and the same initial molecule. So the molecule got later on duplicated. So, if once we get all this, have sequenced this and get all our reads, we analyze it, and what we will do is that for each read, that we transform the whole thing in a big matrix, and this matrix has which way do I try and do it? Let's do it like this. One column for each cell, and one row for each gene. We do the following. Whenever we see a transcript, we get a transcript, we look at its cell barcode, and then we add here a column for this cell barcode if we see the barcode for the first time. And if the barcode has already been seen before, then we say, okay, this is a news, this is another transcript from a cell we already know, and add it to this. So each of these columns here is labeled with one barcode, and hence should correspond to one cell. in each of these genes here, and we get from the other side, because we have sequenced also the actual transcript, so we know which gene it comes from. And you know, the human genome has something like 20,000 cell coding, protein coding genes, and if you also add non-coding genes, then we get something like 60 or 70,000. So we have this. So we have the corresponding number of rows. And in the actual entries of the matrix, we count how often we have seen a read originating from this gene with this barcode. And this is what we then have. And each of these columns is what we call a cell expression vector, which tells us something about one cell. And what we want to get in the end is and figure out what does each of these columns tell us. So the first thing we want to understand is what kind of cells do we have in our cell. And for this, we take, we first want to group all these columns together into groups of similar cells where the same kind of genes are expressed, and then go on from there. And what I now do is I show you a primitive, analysis, how it's usually done in R to analyze this kind of data, and then we can see a bit how this data looks like in practice. And I jump right into the place with this matrix, because that's sort of a starting point where we can easiest see how the whole thing will work. And we will do in some later lectures, we'll talk about how we actually get from the sequences to the mapping. So when all the stuff that I've told you now theoretically starts looking a bit more as something you can have an actual imagination from. Now, in order to do this, when this technique was new some 6 or 7 years ago, most people try to adapt the workflows which were established for the older bulk sequencing method, where we didn't know which reads is from which cell, and came up with a couple of new stuffs, especially the things that people had to think about is how to organize, how to find similar cells, and so on. And quite early on, the lab of Raoul Satir at the Weizmann, what's his first name?
No, they're in New York, no?
I don't know, we keep moving. I think they used to be in the Weizmann. Anyway, there is a lab, there is one guy called Satir, Raoul or whatever his first name was, who then published a R package called Sura, which became widely popular as the standard thing that everybody uses to do it. And as usually with R package, they made a tutorial where they demonstrate the use of this. And I thought as the first thing for today's lecture, we go through this tutorial together. And as I said, we first read fast, not trying to just see where we are getting to, and then we go back and go through all of these steps in detail and try to understand the mathematics that's actually happening there. So here is the homepage of this Sura toolkit. So Sura, for those who are not art historians, was an impressionist painter of French painter of, I don't know, whenever this time was, when the so-called pointillism was fashionable. And he liked to point, to paint, you know, in this shape of making with the pixel strokes. Why the Satir group decided to name the software tool for this painter will become obvious once we see how they typically visualize their data. At least it seemed to have reminded them of that. And yeah, now here they have made these handful little vignettes to demonstrate the different aspects of their data, of the different functionality of their packages. And as you can see, they are by now in version 5, and they have a lot of different things, and we will see quite a bit about this over the lecture, using either their stuff or the competition, which is a package called Scanpy from the TICE group in Munich, who did more or less the same, but not for R, but for Python. And of course, lots of different other things. But what I now start with this thing, which was the very first vignette to demonstrate the very first version of Sura. It allows us, it's a guided tutorial, as we call it, where we have as the name says, 3 cases of PBMCs. For the non-biologist, PBMC stands for peripheral blood mononuclear cells. So peripheral blood simply means blood taken from the vein, as your doctor would usually do when they draw blood. And mononuclear means cells with one nucleus, because in the blood, usually a cell you might think it has one nucleus, but in the blood it's a bit odd. There's a couple of white blood cells which have multiple nuclei, and there's the red blood cells which don't have any nuclei at all. And you use a so-called flow cytometer to sort those away and only use the mononuclear cells to keep things simple. And so we use somebody's blood, just taken from the arm vein, and run it through one of these fax machines to sort down to only the mononuclear cells. Or actually, I think you use a fecal gradient. The biologist, please correct me how you get them. To just sort them by density, and we use those of intermediate density, and now we get them, and we will now see whether we will be able to distinguish the different kinds of mononuclear cells which we expect, such as monocytes, B cells, T cells, and all the other cells. In order to see how this all works, I will now simply follow this thing here by doing it live on my R, and you can see whether I managed to just run this without it not working by doing it. But you don't have to take too many notes, because as you'll see, I'll mainly copy and paste code from here. And see if I can get this a bit larger. And so you can read it a bit later. So I load this server package. Who has never worked with R so far? Everybody knows some R, okay, except for Andreas, so you're creative now. Here is, I load the server package, and now I load my data. Read 10x. Let's see, so here they have put the raw data here, and I've just downloaded the data before this morning. See the downloads. LS, what was it called? HC13. Sorry. Yep. Just have to see what here exactly. That was my last download. PBMC 3K for 3,000,000 of them, and as you can see, this data contains 3 matrices, which, if we have a quick look at them, is here the actual matrix, and the matrix is written down in what we call a matrix market format. It's a sparse matrix, because as we will see in a moment, most of these matrix elements are 0. So I do it the other way around. I first load the matrix and show you how it looks in R, and then we can easily start it. So we're supposed to take read 10x, /downloads /hg, what was it here? Filter gene expression. As you see, this has already been pre-filtered. What this means, we will also have to discuss later. So I read these things, and once I've read them, I get here this matrix. I call it the count matrix because it tells us how often we see each of them. And I show you now how the count matrix looks in the top left corner. And here you see this is how it looks like. These are the barcodes. So these little pieces of sequence which tell us which beat it was. And these are the genes. And the dot means 0. And we can maybe take one random cell out of them to have a bit of a thing how this looks like. So I take just randomly the cell number 763. So the cells are in the columns. And now you see here how for most of the genes, the gene count is 0. I can make a table to see how often we see each count. And as you can see, 31,000 genes are completely zero, 500 genes have one count, 80 genes have two counts, and so on. I have to say this is an old data set. By now things got a little bit better, but overall the data is quite sparse. Another thing which we can do to get a first feeling of how much data we actually have is I take the column sums, so I calculate for each cell how many genes we have in it. And here you see typically it seems to be around 1,500, 2,500. So a typical cell, we see something like 2,000, 3,000 transcripts. With the newer data sets, it goes up to 6,000, 7,000, but it's not much more. An actual typical mononuclear blood cell has around a few 100,000 counts. So as you can see, we get something around a few percent of a transcript we actually sequence, but the majority we are losing. But as will be important afterwards, when we think about the mathematical modeling, this is a completely random process in which cells, which genes we actually get or not. So we will later in our mathematical modeling assume that we have our actual complement of, say, 250,000 RNA molecules, and out of this we sequence at random something like 3,000. And then we ask, what is the expected variance of this sequencing? And we will quickly see that the expected variance is well described by the Poisson distribution, and this will have important consequences in how we deal with this data. For now, I will simply to just see how this looks like. I will take these call sums and I will make a histogram so that we see how this looks like. And you see we have typically, occasionally we have more than 10,000, but usually we have around 3,000 or so. The histogram can sometimes be a bit clearer if I take a decaded logarithm, and now you can see the majority is here. These are 1,000, these are 10,000, these are 3,000, so we have here a mode around 2,500 transcripts which we see in each cell. The next thing which we might want to look at is how similar are two random cells. So I now take one of these cells, the cell number 763, and I take another cell, let's take cell 942, and I make a scatter plot of one against the other. And as you can now see, each dot is here a gene, and you see there's a couple of genes which are high in both cells. There's a couple of genes which are high in only one cell, and so on. And in the end, if we now look at this cell, which has 45, which has lots of counts in this cell and hardly any counts in the other cells, that will probably tell us something what's typical of this cell. But we want to do this in a bit of a more systematic way. So let's go on over to the Serra object. And what I take here is, and I first follow simply the tutorial, which says I should put this all into one big object, which we call a Serra object. It's just one data container which takes all the data, which will contain all our intermediate results, and I give it my count matrix. And then what I will also give it here is these two things here. I tell it that it should only keep those cells which have at least 200 different genes expressed, so there's at least 200 different non-zeros in the column, while a cell which has only 2 or 3 genes expressed at all should be kicked out. And all the genes which don't appear in at least 3 of the cells should also be kicked out, simply to remove stuff which is uninformative. And if I do this, I get this object, which is called an object of class Serra. I will call it 3. And now I get this thing, and now there is some standard analysis workflow. And I skip over a couple of quality control things because we can do this later, and will first normalize it. And normalizing means, so how is this called? Normalization method. This is all the default, so I can just leave it like this. And I write it back. Now I have normalized data. So what does normalization mean? First thing, we have seen that that the number of cells of reads which we get from each cell differs a lot. Some cells have 10,000, others have only 1,000 reads. But we are not really sure that this is very meaningful. And if we later want to compare whether a gene is more strongly expressed in one cell than in another cell, it might be useful to account for the difference in total number. So in other words, what we want to look at is not the absolute number of reads we get from a gene, but what we want to know is the proportion that we say 0.002 of all the reads fall onto this cell and 0.007 fall onto that cell. So if I take, again, my one matrix, which I have here, this one, I can divide it by the sum of all the counts. And now each of my, of course, the zeros stay as zeros, but all the non-zero things will now tell us the fraction, which fraction of all the reads in the cell come from the respective gene. Another thing that you might have noticed is that our data is extremely heteroscedastic in the sense that small counts are quite similar because they can't differ much, while for big counts you have a lot of difference. And obviously, whether this point here has 60 or 50 counts probably doesn't make that much difference in practice, but it seems to be a difference of 10. And we would rather want to work on the ratio. If we say it's maybe in one cell, it's maybe 10% more, when rather it goes by the absolute thing. So we want to turn ratios into differences, and because of this, we take the logarithm of all this stuff. So I take the logarithm. Let's take the log 10. And now, of course, you see all the zeros turn into minus infinity. And now, as a preliminary thing, we do something which is a rather cheap trick. We just add a small number, so that 0 counts turns into 0.0001, and 1 count into 1.0001, and then we get this here. And now zeros turn into minus 4, because that's the log 10 norm. And that's what this thing does here. You see here, that's what they see with this scale factor. That's the 10 to the minus 4 that I've just put. So this here we just divide the total number of each cell and then add 10 to the minus 4 and take the logarithm. Next, yeah?
So just to make sure, you normalize within the cell.
Yes, for each cell we divide by the sum of all the counts in this cell, and then next what we do is we want to find variable features. So we want to find those genes which are especially informative in order to later save computation time and only take those genes which are very and here this is called find variable features. And you see the way how the Sora works. You always run a function on this object and write the result back to the object. And then the object gets some extra slot which now contains these variable features. The problem is you never know where they put them. So they have all these slots. And actually, I can't say to you now, Philip, where they put the variable features. Anyway, it somehow now puts us a list of all the variable features, and we can also plot them here. And here, as you can see, each dot is a gene, and the x-axis is just the mean over all the genes, and the y-axis is the variance. So if we take our log transformed matrix now, we calculate over each row, so for each gene we calculate the average and we calculate the sample variance, and then here we plot the variance against the mean. And as you can see, there's a couple of genes which have high variance, so they differ a lot from cell to cell, a couple of genes which have low variance, so they are the same in all the genes. And if we as our first goal is to distinguish what different kinds of cells we have, and the top ones are more informative than the bottom ones. We want to stick only to the top 2,000 of these and not use these 11,000 which are all overplotted here and which are more or less constant across the cells. Then what we will do next is when we do a PCA, and I will postpone the explanation why we do a principal component analysis to afterwards. But what this does is, I can roughly explain it at the moment, what we want to get at the end. Maybe it's easier if you already know what we are heading at, so I quickly jump to the end. This is where we will get in 5 minutes. Each point here on this so-called human plot is one of our 3,000 cells. And we have established a distance matrix between cells. We will now in a moment find a way to take any two cells and ask how similar they are. And afterwards, we will visualize them here in this two-dimensional manner such that whenever two cells are similar, they are placed close to each other, and where they are very different, they are placed far away. So all these cells, they are sort of similar to each other, while these cells here are very different from these cells. And that already allows us to see that our cells roughly fall into 3 different kinds. And after a bit of further looking, we will then realize that these are the B cells, these are the monocytes, and these are the T cells. And then if we look into the T cells, we will furthermore notice that there seems to be kinds of T cells which are quite different from those here. And we will realize these are the naive T cells, these are the T cells which have already learned to recognize one specific antigen. These are the cytotoxic T cells, and up there this actually sits with the T cells, but some cell type which is similar to T cells but aren't namely natural killer cells. In the same way, the monocytes will fall into different subtypes of monocells, etc. And in order to do this, our crucial point is we need a way to compare any two cells and say how similar they are. And the big issue that we have there is if you look at this picture, and now you want to say, are these cells similar or not? You might look at those strongly expressed genes here to compare the two cells, while the blob down here at the bottom of all these genes, which are only 2 or 3 counts, they are not very useful to compare the cells per se, because they are subject to a huge amount of Poisson noise, in the sense of, you can imagine, if you would have the very same cell twice, and you sequence them twice, you would still not be surprised to see 3 counts for the gene in one copy of a cell and 4 counts in the other copy. So whether you have 3 or 4 or 5 counts for an individual gene doesn't matter. But if you were able to have hundreds of copies of the very same cell, then you could average over all these very low numbers, and you would see a difference. Unfortunately, we cannot have each cell only once, so we can't do that. But what we can do is make use of the fact that all genes are correlated. We know that genes depend on each other. A whole couple of genes might be regulated by the same transcription factor, so they are all up together or they go all down together. Or a couple of genes might be specific to B cells when rather to T cells. So there is certainly dozens of genes which have only 2 or 3 counts in B cells but 0 counts in T cells. If we would know these things, then we could just average over these dozens of genes and get one number, which clearly distinguishes a B cell from a T cell by being something like 4 or 5 B cells and 0 in the T cells. But in order to find such things without doing any individual, without already putting in prior biological knowledge to do this automatically, we can use principal component analysis, PCA. Who knows what PCA is? Okay, most of you know, and we will. Who knows how PCA is calculated? Good. This is what we can do the next time. That was my plan to start off. As I said, we want to go into the details of mathematics, but as a sort of initial example of getting into the details, we look a bit at how a PCA is actually calculated. But for now, let's simply do the PCA. And because PCA is a bit is a bit computationally intensive. We don't run all our counts, but only on those counts, on those genes which have a certain variability. We already know that probably those which are down here at the bottom, which are more or less constant over all the genes, they probably are not too useful to be part of the PCA. So what we can now do here is I can say run this PCA. Where are we? Run PCA server and write the result back. And data has not been scaled either. Before it wants to run the PCA, it wants to scale the data in the sense to make sure that each gene has, from each gene, we subtract the mean and divide by its standard deviation, so that each gene is more or less standard normally distributed. When we discuss about what the PCA actually does, we will see why this is a standard treatment, that whenever you do PCA, you must center first, so you must subtract the means first, and you should divide by the standard deviation. For now, let's just do it and then run our PCA, and I think by default it runs only on these 2000 most variable regions. And once we do this, we get this we calculate the principal component. And to remind you all what the principal component means is, we now have a new vector which looks like this. Sorry, add reductions dollar. So this server is terrible in the thing that it has all these complicated slots, and you always keep, by now I have remembered that this is here, and now you see how this works. For each cell, we now have a principal component vector. So this cell, the cell with this barcode, is now described by these principal components. So before, each cell was described by a vector of some 30,000 gene counts, most of them being 0. Now each cell is described by a vector of 30 principal component scores, which are not 0. And I can plot, for example, now the first principal component against the second. And we can already see that this helps a bit, that now our cells clearly have already split into these 3 blobs. So what does a principal component do? What is a principal component? A principal component is a linear combination of our initial vectors. So something like PC1 is now a sum of gene 1 plus gene 2 plus gene n. And where this here are the log, normalized, and scaled expressions. So you remember we first had the count, we divided by the total sum, added 10 to the minus 4, took the logarithm, when we subtracted the mean over the whole gene, divided by the standard deviation, and then we got some number, which is this one here. And now let's call this here A11, A12, A1n. So when to calculate our first principal component, we add up the expression of all these genes, but before we add them up, we multiply each of these things by some number. And these numbers are now chosen such that that we get a combination, that each principal component is a correlation of useful vectors. So for example, as I said, we will find many genes which are correlated with each other because they are related to the same biological function. And now the first principal component, maybe that one picked up all the T cell genes. So when all the genes which are typical for T cells get a high value, a positive value, all the genes which are not typical, which are never seen in a T cell, get a strongly negative number, and all the genes which doesn't tell us anything about whether it's a T cell or not, give us the gives us gives us something around 0. So what I can do, for example, is I can go here at the reductions, $PCA add, and here these are called the loadings, which now tells us about for each gene how strong the loadings is. So these are now the loadings for the first principal component. Just say comma 1. Here we get them. And as you can see, I plot them. Most of them sit around 0, but a few are strongly positive or strongly negative. Before it has even told us which ones these are, here you see here it here it told us strongly positive are these ones here, strongly negative are these ones. An immunologist will probably have one look at it and can already tell you which genes are what's separated here. I'm not an immunologist, but by now I've done this tutorial often enough that I immediately see CD79A is the B cell receptor and it's positive here, so PC2 probably distinguishes the B cells from the others. And hence, those which are strongly positive for PC2, these are probably the B cells. So this is what the PCA does. We call it a dimension reduction. Before we had this 20, before each cell was described by this 20-dimensional gene vector, now it's described by 30 numbers or 20 numbers or so. And now it's much easier to compare two cells, because if I now take any two cells and just compare these 30 PCA numbers, I usually will get either they are very similar or they are very different. And that's what I can now do. You can already see from these first two principal components, this cell and that cell, they are probably very similar, while this cell and that cell, they are clearly very different. So if I now compare any two cells, I can clearly say whether they are similar or different. Let's try this quickly by taking here not two components, but let's compare cell 345 with cells 378, comma missing, and this now, and now in order to see whether these cells are similar or not, we check the correlation between these two, and we probably would find a strong correlation if they were similar and a weak correlation if they aren't. I could now try this with a couple of different ones, and eventually I would probably find two. These here, you see, they look sort of correlated. And what this now allows me is that next I can now calculate a Euclidean distance by taking for any two cells the principal component vectors and just subtracting them from each other. And after I subtract them, you all probably know, who knows how a Euclidean distance is calculated? Okay, so you all know we just add up, we take, for any two cells, we take the differences between these two components, square them, add them up, take the square root. Using this, we will get now a score which tells us for any two cells how different or similar they are. And now we calculate a t-SNE or a UMAP. Let's take the UMAP, served. And what this now does is it calculates.
Need the KNN
in the UMAP, yes. Right. Let me sneak at my tutorial, perform dimension reductions. So here you see in the tutorial, we just looked at this plot. Here they've also made a heat map where each gene, where each column in this heat map is a cell, and each row is one of the genes which are very positive or very negative in the first principal component. And we can see that the PC neatly separates our cells into two blocks, which is either strongly positive or strongly negative. And in the same way, we can look at all the other PCs to figure out what these PCs actually separate. And we can now figure out, if we were to look at what is interleukin 32 and interleukin 7 receptor and C2, I think this is all T cell stuff. And we can compare them. So what we now next typically do is we do this find neighbors. What finding neighbors does is it takes, tries to find for any cell 30 other cells which are close to it. Close in the sense that they have small Euclidean distance to this cell. And if I run this, I get now, let's see here, neighbors. I don't know where I put that. So I always find this very frustrating in Further that all these functions just write the result back into the object and hide it in it somewhere, which is why what we will do next week, no, in 2 weeks, because next week is 1st of May, we do all this, but only using standard R function and standard numerical libraries without using any specific thing, so that we really see what's going on instead of doing this. Today, I just want to run through it so that you can see what the result will look like, and next time we will replace each of these functions by something where we can actually look inside and understand what the math is doing. So what we should get here, and I don't know where I put that, is that it makes us a table which tells us, for example, cell number 326 has the following 30 close neighbors. Cells 13, cell 27, cell 312, and so on. And it tells us these. And what we then can do is run the UMAP function. What is find clusters? I don't want to do that. Tell the UMAP function it should take the it should take the 30 PCA dimensions. And now try to find and now make this UMAP plot. I have the other way around. UMAP plot, and make this plot that we have already seen, where each cell is now arranged like this. So what's happened here is this is an optimization problem. So the optimization, what we want to do is we want to put each cell on this 2D plotting surface such that it's close to its 30 neighbors and far away from cells which have a huge distance to it. And there is 2 common means to do which called t-SNE and UMAP. I've showed you here the UMAP, but I will explain the t-SNE because it's easier to explain. And there I will always first give you only the gist of it, and the details we will also see another week. We come up with what we call a cost function, which tells, which checks how similar 2 cells are in PCA space and how similar they are here on UMAP space. Actually, maybe the easiest is if I try to demonstrate this quickly. So this, now you can see if I manage to do this without. So we have 2,700 cells. 700 sample dot int. So now the other way around. So this is to pick 10 random cells. Now I will pick 100 random cells, and I will actually allow the thing to pick the same cell twice. So let's call this random cells 1. And let's take 1,000 cells so that this works properly. And I do it again. I call this random cells 2. What I will now do is I calculate the distance between the first set of random cells and the second random cell. So I want to calculate how similar the first cell in this list is to the first cell in that list. And I will calculate this twice. Once I will calculate how similar they are in PCA space, and second, I will calculate how close they are to each other in UMAP space. So let's do the UMAP space first. So I take my third object, take the reduction, take here the UMAP, cell embeddings. By now I've learned how to get this. And if I now take, say here, I want the first cell, then you see I get the UMAP coordinates of these cells. And if I say R cells 1, I get the UMAP coordinates of my 1,000 random cells. We do this again with the other 1,000 UMAPs random cells. Then I get another set of UMAP coordinates. And I want to see how distant are they from each other. So I subtract one from the other. Now these are the differences between one random point and one other random point. And I want a Euclidean distance, so what I do is I take this, square it, and sum up the two columns. So row sums of this and that. And now you see for each pair of cells, I see here the distance. Actually, for copper Euclidean distance, I should take the square root of that. Everybody with me so far? And now I do the same for the PCA. For the PCA, I just do here, instead of UMAP embeddings, I say PCA embeddings. So as you can see here now, this here now gives me for each cell its PCA coordinates, and here again the PCA coordinates. And this gives me now the distance of each cell pair in PCA things. And what I now do is I plot one against the other. So, and what we can see now is how well this UMAP actually has worked. We can see that the x-axis, for remember, is our UMAP distance. So for each point here is a cell pair, and this here tells us how far the cells are in the UMAP, and this here tells us how far the cells are in PCA space. So in a way, we can say this here tells us how different the cells really are and how different they appear on our two-dimensional plot. And as we can see, the first thing has worked quite reasonably well. All the cells which are similar, so which have a real distance below 20, also appear here. But what hasn't worked well is once the distance here is above 5 or so, we cannot really conclude whether they are somewhat different or very different. So this tells us a bit about how well this works. What we have done a while ago, a PhD student in my group, hey, where's Sleepwalk? Library Sleepwalk, let's see. Oh no, I don't have it installed. Just give this a second. So now server reductions UMAP. So I have it in a, you see in a second what this thing does. PCA. Here you now see a graphical description where whenever I put my mouse on a cell, it will color the surrounding cells according to their distance in PCA space. And what you can see in this manner is, let's make the scale a bit better, is here if all the green cells are cells which are really similar to the cell under my mouse, all the purple cells are cells that are somewhat similar. And this now allows you to see how well this has worked. So here, for example, you can now see that all those cells which are now dark green, they are more or less the same. By all those cells, we stick a bit out here, they are probably not so similar. And you see on top there's a couple of cells which look similar at those at the bottom, even for where far away. By a couple of cells here which seem to be closer are not so far. This is not surprising. What we've done here is a double dimension reduction. So we started off with our 20 or 30,000 dimensional feature vector, which had one number for each gene, most of them being 0. And we thought this thing is too clumsy to work with, A, because it's too long, and B, because it contains too much Poisson noise. And then we used the PCA to condense this 30,000 dimensional vector down to a 30-dimensional principal component vector. And we had the feeling that this actually improved things. Now, the distance in this PCA space is more representative of the actual biological dissimilarity between the cells than the original distance, because we averaged out the Poisson. But if we go down again from this 30-dimension to only 2-dimension, we have overdone it. And with only 2 dimensions, we now lose information. The reason why we do it anyway is simply because 30-dimension is so hard to plot, unless somebody invents a 30-dimensional screen. So we use this second dimension reduction from 30 to 2 in order to get a rough feeling. But when we do this, we should always keep in mind that this is a crutch which only helps us to visualize it somehow for our 3-dimensional brain. But we do miss things. This is why for everything which follows now, our rule will be that our calculations will always be done in this 30-dimensional PCA space. We will imagine that this 30-dimensional space is sort of a representation of a real state space, and the 2-dimensional things not so is just a crutch. But nevertheless, we can imagine that if we were able to visualize this 30-dimensional space, we would probably find one cluster or blob of cells which are quite similar and which are maybe this blob here, and another one like this, and a third one like this, and they are well separated. And what we now want to do is to find clusters of cells which are very similar to each other, to simplify our thing, because we now here have 3,000 cells, but actually we are not interested in individual cells, even though we talk about single cells, because if a cell appears once and when we see it never again, it doesn't tell us any biology. In science, we are always interested in reproducible stuff, so we want to generalize things. So what we want to know is actually want to find classes of similar cells so that we can average over these classes and then say, okay, this average, that's probably holds. When we do this averaging, we will have to be a bit careful, because at the moment all this is one blood sample. We will check whether it's also true for a second or third or fourth blood sample. But for now, the first thing we want to do is to group these cells into what they call clusters of similar cells. When we call it clusters, there's some mathematical intuition into it, which got somewhat a bit distorted on the way from math to biology. So something that especially the mathematicians in the audience have to keep in mind is whenever you work interdisciplinary, technical terms get a change in meaning and something gets lost. So what a mathematician or a data scientist would call a cluster is what you imagine. We have something like this here. This is, each point is some cell, and this is some 2-dimensional feature space. And when we would say, okay, here we have a cluster of cells, because all these cells are within a small region. And here we have a cluster of cells, and these 3 cells, they sit in the middle in between. We might also have further off something like this here, and we would now wonder, is this a cluster or not? On the one hand, the distance between this cell to that cell is no larger than the distance between any 2 cells here. But on the other hand, all the cells are much more loose here than here. So maybe we should also call this a cluster. So we will have a bit of an issue between how we use the density, whether the cells are spread out in cell space or not. Biologically speaking, this means some cell types might be very tightly regulated, but all the cells are exactly similar and they sit very close to each other in PCA space. And other cell types are not that finely regulated and they spread out more because there's more variation from one cell to the next. For example, immune cells which are activated and active in combating an infection, they will be quite spread out because each cell tries in another state of tries something different to combat the infection, while skin cells which just sit there and do nothing, they are probably not that spread out. So, and we want to find such clusters. So what we do here is one thing that where the word clustering, as I said, has two different meanings in actually already two different fields of mathematics. Is here we might call this a cluster, and this a cluster, and this a cluster, and this one here we don't consider part of a cluster. Another way what we could do is that we make a partitioning where we partition the whole space and say everything in this partition belongs to that cluster and everything in that partition belongs to that cluster. And that's what the people in single cell usually do. Every cell is forced to be pushed into a cluster. If you have a cell which is completely different from everything else, it will also be pushed in. We will come later in this term to talk about why this is a problem and how one could do it better. But for now, we simply do it how everybody does it. And the reason why everybody does it that way is because the Server people implemented this in the Server package when the whole thing was new and you just wanted to get some result. And there's a lot in this whole field which people just did because they always did it and didn't really reflect. So let's see. And the way how we do this is first from the point of this thing is simply that we say, well, you guessed it, find clusters. And after we've done that, it tells us that it found 11 clusters. And I can, if I now run the UMAP plot again, then it shows me each of these things by clusters. And as you can see, this sort of makes sense. Let's make this halfway square, that here it has really noticed that these are all quite similar and are one cluster, while we've noticed that these cells have still some difference between these and these, so that it split it into several clusters. You can also see this in my sleepwalk thing, where you can see here there is quite some difference between these cells are quite dissimilar from those, while if I go to a thing up here on top, I can sort of put the whole in one uniform green. So, and what people now do next typically is that they want to understand what's the difference, what are the three clusters. I already told you, one is the B cells, one is the T cells, one is the monocytes. And the way how they suggest that is that we say, let's start with this single cluster, cluster 2, as you can see, and ask which genes differ strongly between cluster 2 and all the other cells. And they call this the find marker gene functions. Find markers, I say Säure, and then I have to say, take all cells from cluster 2 and compare them with all the other clusters. And then it runs just some, I think, a Wilkinson test or so, comparing the expression strength of all the genes in this cell against all the other genes, and then gives us here those cells with the highest p-value. And if I go here at the beginning of this list and use my biological intuition, we see here immediately there's this gene CD79A, which is expressed on a log2 fold chain scale, so on a log2 ratio 2 to the 4 times higher in these cells than in the other cells. In 93% of those have at least one count in it, while only 5.2% of the cells in the other clusters have more than 0 counts for this cell. So overall, we can say that this cell is much stronger in one than the others. They also have this function which we call feature plot for this, where you can take such a such a feature and ask it, color it by log expression, and you can see CD79A really seems to put this one out. So what do I do next? As we have seen here, the top of the list was MS4A1 and CD, also known as CD20. So anybody of you who's ever done immunology knows what CD20 is. It's the standard surface marker that we use to find B cells in facts. And CD79A is the alpha chain of the immunoglobulin Fini. Which one is it? There was always here. Here you have a B cell receptor, and you see this is CD79A and B. So this is your typical thing. If you have very diverse cells like this, what you can then do is, in this way, find the cells. In the same way, I could now look, of course, go two ways. I can, if I already guess there should be T cells, I can take the T cell receptors or the epsilon chain of a T cell receptor and plot it, and we'll immediately see, okay, these are all T cells except for this little tail at the end, which might not be T cells. And in this manner, we can now go through either all the marker genes, which we remember from the last time we listened to an immunologist talk, or the other way around, we can use these fine clusters to figure this all out and compare things. So, for example, if I now want to know what is the point of this little thing here which doesn't express CD3A, so this cluster number 8, I could, of course, say compare the cluster number 8 with the cluster with all the other clusters in here, which is, if I look at the colors correctly, 3, 6, 1, and 0, I think. And it will probably tell me genes which a immunologist would immediately say, obviously, this is T cells versus natural killer cells. So this is one part, but of course we haven't learned any new biology from that. So far we have just learned how to find different cell types. So we should see a bit of what we can do next. So the big things which now come around next is, let's imagine we would now have blood samples from 5 healthy people and 5 people with some disease. And we want to know what is the difference between the T cells in the healthy people and the T cells in the patients. When we might we need to now not only compare cells within a sample, but between samples. Another thing that we might want to do is, these were 3,000 cells. By now we have much more. Here I put up a paper of 2 years ago, which wanted to see the development of the mouse brain, and they have 500,000 cells. So let's have a look here at this big first picture. This is where T-SNE or human plot. Each point is a gene. You can hardly see the individual points in this thing. They have put here these little surrounding plots to show you what happened. So they took mouse embryos of different ages, and as you can see, these are the cells of the 6-day-old embryos, of the 7-day-old embryos, of the 8-day-old embryos. Now comes green of the newborn, and we can really see how our cell types differentiate. In the early embryo, there's only one small type of cells. When they later expand into these many cells, and when these cells, around 10 days, they split into 2 subpopulations, and around 16 days, this subpopulation has split again into this and that. And when they later tried to distinguish this, they found out these are the radial glial, these are the excitatory neurons, these are the inhibitory GABAergic neurons, these are the forebrains, these are the others. So we can see how cell types spread out. After all, we all start with a single X cells, and then in the end we have hundreds of different cell types, and we can see here smoothly how they change. And this will require us a lot of mathematics to try to understand how we can now understand this branching and follow the evolution from the simple germinal layer cells to the fully differentiated neurons. You can imagine that in our high-dimensional feature space, we now don't have individual blobs, but what we instead have is elongated long stripes of cells, which start somewhere in feature space, and they change, and they branch into different things, and we want to follow them. So for the mathematical side, we want to find curves through this 30-dimensional PCA space, which starts somewhere and goes through it, where we know this. And when exactly does it branch? How do we figure out which genes cause the branching, which genes separate, mark the difference between, say, a neuroblast, which is going to be excitatory, and the neuroblast, which is going to be inhibitory. We want to understand what regulates this, what the biologists call fate choice. And for this, we have to identify where in feature space do they split, how do we bring the cells into the right ordering, starting from the early cells to the late cells, etc. And this is quite beautiful math, because we do then a lot of geometry in our high-dimensional space. We want to put in curves, we want to estimate manifold shapes, we want to understand the whole topology of this. Is it branching? Are there loops? Do things merge back? When do things change? And for all these things, we will need to use mathematical tools which allow us to explore this. And we really will need to combine a biological knowledge with this mathematical expertise in order to understand where we are going there. So now I've got 90 minutes, which might be about enough. Maybe we should have made a break in the middle, but next time. So what I want to talk about next time is that we understand a bit more what's going on in this feature space, and to this we will look a bit more in detail what the PCA actually does, so that we can later understand what the PCA does before. So as for me to help me prepare, let's first start with a question. Who of you knows what eigenvector is and remembers how an eigendecomposition is calculated? Okay. Most of you, good. Then let's see how far we get with that, and do a bit more math. So today we've just looked at funny pictures or colorful pictures. At least this one here I think is very colorful. And next time we will do some actual math. At least start with something familiar like an eigendecomposition, and then from that go to something else. We will also probably get into things like alternatives to the PCA with neuronal networks and autoencoders, and let's just see how far we get within our one term. I've noticed that next Monday is May the 1st, which is a holiday, which implies that we only meet again in 2 weeks. Yeah, okay, then I thank you all for coming. Please let me know over the time about where I should lay the focus, so you can help me steer it a bit, whether we more go to the math or to the biology side or to the computer science side. We will actually try to maybe implement something ourselves, like I thought we could make with UMAP or TSNE, not the UMAP, but we can just program the TSNE ourselves. Oh, by the way, who knows Python? Good. Because for most of these things, if we actually want to do something ourselves, it might be easiest to work in Python rather than R. Okay, then see you all in 2 weeks. Yeah.
So for me, the motivation was all over the place today, because there were a lot of new terms. So is there a script or something that I can go through to understand the biology part, or a reference point maybe?
That makes it a bit difficult, because of course we now have exactly, our non-biologists, how much biology catch-up do you need to? I guess you're the only one who's really completely outside biology. You as well, okay. For me to now write down a script about the biology just for the two of you might be a bit, so we can just meet the three of us and quickly go through the biology part, because that might be easiest. So let's just meet the three of us, and I tell you a little bit the biology that I've learned over time, what transcription and translation and central dogma, and all the stuff which would be too boring for all the other people, I guess.
And when do you wanna do that? When should we meet? Because next time when we are meeting, I have exams, so next Monday I won't be here.
Yeah, let's do it next week. What was your two names again?
Purusharth and can I?
Yeah, just write it down.
Can you also write my name at
Okay, perfect.
So next Monday I won't be available with anything after that.